Hello everyone, and welcome to the second part of the Chapter 18 overview lecture here, where it's just a complete review. It's very, very short. I'm covering essentially 4 to 5 hours of lecture in about one, so you need to have a good background knowledge of this. Otherwise, this isn't going to make much sense. And if you get that kind of overwhelming feeling that, hey, this isn't making sense, it's because you're not quite ready for this. Otherwise, if you do get that, it should be a lot of as I'm going through it, and again, I'm going to go through it very quickly, but as I'm going through it, you should be nodding your head most of the way through it, saying, yeah, this all makes sense, and then anywhere where you're kind of like, oh, I didn't realize that, you should be jotting that note down, and then that's what you're working to get clarified. All right, so that's the point of these overview or these review lectures. They're not the lecture you should be listening to first. I'm going to cover lectures 3 and 4 in this. I'm going to cover it in about 35 minutes when normally each lecture there takes me close to an hour, especially lecture 3 being one of the longest. All right, but let's get into this lecture here. As usual, you can pause, review these. You should try answering these right now and see, what do I hit on when I discuss these? Do you hit on the same main points I do when I go through these? All right, so thyroid gland, you should definitely be able to recognize this on a model in the lab or a histology slide very similar to this. Usually I use something that's, you're going to see a lot more of the surface area of it. This is kind of zoomed in a little too much. I would zoom out a little bit to give you a little bit better of a view, but this is still a great image. You can see clear, simple cuboidal epithelium. Should be very obvious, all right? So when it came to the thyroid gland, this is all of the simple stuff. I am not going to spend too much time describing these things. You should know that thyroid follicles are these circular-like structures, and in fact, you should know that the cells here are called follicle cells or follicular cells, each of these that form a follicle space. Now, it's rare that I ask what is the open space here. If I do, that's the follicle cavity, but usually what I'm asking is usually what's this pink substance. If you remember, the pink substance represents the colloid, and that's hopefully something you already remember, and that colloid is filled with TGB. In fact, that TGB is going to get converted to globular T3 and T4 at some point that we know gets formed in here, and then it gets brought back into the cells to be chopped up into individual T3 and T4, which is a concept that I lectured on. So all of this so far should be like, yeah, I remember these concepts, even though I'm not there on technically the lecture slide, right? So far should be things that you were kind of obvious, kind of recognizing. And then again, when we're looking at these, make sure you know these pockets where we don't have follicles, that these are all the C cells. And if you remember, C cells secrete calcitonin. So when you look at this, usually the question that I ask is, what is this a histological slide of? Obviously, all of these are fair game. And during class, I would have to tell you that. I'm talking like this purple region here. What is that portion called? Because obviously, this is the parathyroid gland. All right, so you'd say thyroid gland. I'd probably use one of these other images. And then I would ask you, I can easily ask, what are these cells known as? These would be the follicle cells. I can easily single out the C cells and ask what they are. I can easily ask, what hormone do either cell type produce? Now, obviously, the follicular cells are going to be producing T3 and T4. The C cells are going to produce calcitonin. I can ask, what is the pinkish substance? That is the colloid. So you should know the colloid is the pinkish substance there. Kind of extra credit-wise, I might point to what is this. That's simple squamous epithelium, so that's a blood vessel. That would be on the extra credit side. All right, but also I can easily ask, what type of epithelial tissue is this? And you should know it's simple cuboidal, because you can see it's a single layer of simple cuboidal cells that form those follicles, right? So that was kind of the major points that you should be walking away with the histology. Now, remember, what causes thyroid gland to produce more thyroid hormone was the release of TSH. So TSH, if you remember, was a pituitary peptide hormone that stimulates iodine absorption and T3, T4 production and secretion. So you should know TSH at this point. You should definitely know it's peptide hormone. Again, TRH is what leads to the release of TSH. And you should know what causes TSH to be released. It gets released when our blood levels of T3 and T4 are below the normal range. And so if we get low levels of T3 or T4, or if we have low levels of TSH, so if TSH is a problem, any one of these can result in low physical sluggishness, right? Because this is our main normal metabolic hormone. So if TSH is low, that means T3 and T4 are going to be low. But even just saying, hey, T3 and T4 levels are low, you should know it can cause physical sluggishness. Now, what stimulates TSH, or if I had a question on the exam, hey, the blood levels of T3 and T4 are low, what would be expected? Well, symptom-wise, physical sluggishness, or if I ask a question about them being low, how would the body likely respond? Well, it would probably lead to TSH being stimulated, and TSH should start the process of targeting the thyroid to start producing more T3 and T4. So make sure you know your loops is basically what I'm saying. But ultimately, you should know T3 and T4 is important for development, especially the skeletal, muscular, and nervous systems. All right, if TSH was high in the bloodstream, but the levels of T3 and T4 were low, what would that indicate? And if you remember, this was one of the lab questions. It's one of the lab sheets that I'm going to hand out. If you don't have it yet, you'll be getting it. Most of you are probably watching this video after the course has begun, so likely I've already handed that out. But this would indicate that the thyroid hormone the thyroid gland has a TSH receptor issue. So the thyroid gland has some issue responding to thyroid stimulating hormone. Okay, so understand it's probably some issue with the thyroid gland, just not responding to that hormone. That's why this hormone is high, but T3 and T4 remain low because it's literally not responding. So those are the concepts that you need to be able to work through and kind of understand. All right, but remember, thyroid hormone targets most cells. It binds intracellular receptors because it's lipid soluble. And then notice its function was really energy related and regulation of body temperature and development. If you got these three down, anytime you think thyroid hormone, just think of those three things. Normal metabolism, regulates body temperature, normal growth and development. That's it. Hopefully you know there's an important iodine. Now, you might not know it because it's very rare for us to have this in countries where, we add iodine to salt, and obviously I just gave away the answer. Iodine is important because we add it to salt, and so that's why it's very rare that you're going to see in developed countries these goiters that can exist, which is literally an enlargement of the thyroid gland. If you remember, during T3 and T4 production, iodine absorption is a must, and if that iodine doesn't get absorbed, we can't actually form T3 and T4, and if we don't form it in the actual colloid, well, then we can't reabsorb and bring in that globular T3 and T4 to break it down, so it just stores in there, and so your thyroid gland just continues to fill and fill and fill with it fills with the thyroid globulin, but never forms T3 and T4. Like I said, on exams, I list them as T3 and T4. Maybe as a extra credit, you would want to know T3 is triiodothyronine and T4 is thyroxine. But I really don't test on this besides kind of extra credit. And you can always list, if you're trying to list, if I ask what hormone is secreted by like follicular cells, listing it as T3 and T4 is acceptable. All right, but this is really not necessarily test material. This is telling you what thyroid hormone does more specifically, and that's why it regulates metabolic activity, which really tends to be related to oxygen. Okay, it's usually oxygen, red blood cells is oxygen, so, but nothing major testing there. Now, we continue to wrap up. We move into thyroid production, and as I said, you should really know, you should be able to list these steps. Now, you don't need, if you notice, I have 8 steps. The book only has 7. I don't like how they broke them down. I like to break down the steps into a little bit more sub-steps or individual steps. And so really what happens is TSH, technically, if you wanted to add a step, thyroid stimulating hormone binds to the receptor. That causes iodine absorption. So step 1 is iodine ions that are absorbed. Step 2, follicle cells synthesize thyroid globulin or TGB. Then we combine those. So remember, the first 3 steps are getting your ingredients towards the lumen. So bring in iodine, have thyroid globulin, TGB nearby, combine them, and then we ship them. So the iodinated TGB gets shipped into the follicle cavity or into the colloid. So once within the colloid, we know TGB undergoes its reactions and becomes globular T3 and T4. If we don't have iodine, we can't form T3 and T4, so this TGB just keeps building up in here. But once we get that formed, we endocytose it, bring it back into the cell. So we take that globular T3 and T4 and we start chopping it up using our lysosomes. And now that individual T3 and T4 diffuses into the blood and binds to a globular protein. Now, this is kind of the simplified method. If you're not feeling good with all these steps, this is the area I would skip. I wouldn't spend too much time on it. I would focus on really getting these steps to memory. And then the other things that I can individually test on is like what stimulates iodine into the cell? Well, it's the binding of TSH. Where does thyroid globulin get iodinated? It gets iodinated at the apical surface within the follicle cell. Where does globular T3 and T4 form? It forms in the colloid. Where does individual T3 and T4 get formed? It gets formed after being digested by lysosomes within the follicle cell. Okay, how is T3 and T4 transported? It's transported bound to a hormone, bound to a transport protein. So T3 and T4 are transported bound to a transport protein. Those are kind of how I can create individualistic questions. Now, definitely expect to see the full steps on there. And obviously, if you're taking the exam, I'll give you a hint. If you are taking the exam and you know the steps, well, anytime I ask any of the individual questions, hop back and forth between the two, the one that has you list these in order, and then hop back to the individual questions, and you should be able to work out what is the correct answers for them. Now, this is where we started getting into more application. So if you know what the hormones are supposed to do, you should be able to say what the body is, or how the body is going to be impacted if we have too high or too low of secretion. And so obviously with these imbalances, we can have hyperthyroidism, and when we have the word hyper, we think of children with lots of energy, right? So hyper means lots of. And so if we have high levels of thyroid secretion, well, we're going to have high energy levels. We're going to be obviously, if you have high energy levels, that means you're going to be utilizing energy sources and breaking things down. So you're going to probably have weight loss. You're probably going to be sweating a lot because, again, lots of heat, right? Thyroid with heat. So if you just think about it, you should be able to come up with these sort of things. What is associated with high energy? Well, rapid heart rate, high blood pressure, all right? So think about that. It should be pretty easy, in my opinion, to identify thyroid hormone symptoms. Weight loss is going to be one of the major ones. Increased metabolic rate would be something I definitely mention. All right, and then on the flip side, having low levels or hypothyroidism, you should just think, hey, if we don't have the normal metabolic regulating hormone, how is that going to impact us? Well, low energy levels, low appetite, increased weight gain. So kind of the exact opposite. So you should be able to note these. They're worth studying. I do like testing on these anytime we have dysregulation to anatomy and physiology. I like testing on them, whether they're some sort of disease or, in this case, high and versus low levels of secretion. Again, I pointed out the C cells, but the C cells, you should note, are, again, where we don't have a colloid. So this is an area that's a collection of C cells. And you should note that they secrete calcitonin. It's not hard for me to ask on a lab exam, hey, what are these cells right here? Oh, they're C cells. And then the very next question will be, based on your previous answer, what kind of hormones do those cells secrete? Well, they secrete calcitonin. What's calcitonin do? It regulates calcium levels by causing an inhibition to the osteoclast, i.e. slowing down the release. But the big focus is it stimulates osteoblasts. And the way I remember that is calcitonin, I think calcium in, calcitonin in, calcium in. Calcium into the bone. And so the bones uptake it from the blood, and therefore they cause a decrease in the calcium in the blood. Now, I like testing on this from various levels. So you really need to understand calcitonin causes a net decrease in blood calcium. How does it do it? It stimulates the osteoblast, and by stimulating the osteoblast, they are absorbing the calcium from the bloodstream and using it. At the same time, it also targets the kidneys to actually excrete calcium to get rid of it, because again, the net effect is to decrease calcium in the bloodstream. All right, so it's secreted when calcium levels are high in the blood. All right, parathyroid, you should be able to recognize this dark, kind of purplish. Usually I would use, if I was going to test on it histologically, if you remember we had, I think it was, sorry, it's that image way back here where we had this. You should know that that's the parathyroid. That's how I can test on it histologically. All right, but when we go through this, just make sure you know that parathyroid is secreted when blood calcium levels are low. Obviously, it's to increase blood calcium levels. The way you would do that is to target the osteoclast, because remember, they break down bone, and by breaking down bone, they're going to be releasing calcium into the bloodstream. And then I brought up the terms antagonistic and synergistic at the last lecture, lecture number 4. But at this point, you should remember that this is considered calcitonin's antagonist, and you should know those terms and link them together. Obviously, if this targeted the osteoclast to break down bone, that's increasing calcium into the bloodstream. And at the same time, we can target the kidneys again, but we're going to cause reabsorption at the kidneys. So instead of the kidneys pushing calcium out, they would produce proteins that reabsorb the calcium instead of letting them just kind of form in the urine. Also, parathyroid will stimulate calcitriol. Make sure you know that calcitriol does stimulate the osteoclast, but you need to note that it's different because calcitriol does not target itself because the kidney, it's released by the kidney. So instead of targeting the kidneys for reabsorption of calcium, it actually stimulates uptake of calcium via digestion. So it increases calcium via digestion. Hence, if you remember, calcitriol is associated with vitamin D, you would understand that that's why vitamin D is put into milk. So it's when you consume the milk, you can actually ingest and absorb a lot of that calcium better off. All right, as I've mentioned, these figures, you really should study them, but you need to know your main points of calcitonin, PTH, calcitriol. I definitely test on them from various levels. I can easily ask, which of the following hormones is released when blood calcium levels are high? Which of the following hormones targets the osteoblast? Or I would say, which of the following hormones stimulates the osteoblasts, leading to, and I might even continue and say, stimulates the osteoblast, leading to a decrease in calcium. All right, but notice, I have lots of ways I can ask on it. I don't have to be as detailed. I can be less detailed. I can say literally, which of the following hormones stimulates the osteoclasts, and you would need to know PTH and calcitriol target the osteoclasts. Okay, so expect to kind of see that as we move along there and really note those things. Obviously, the first two parts of this lecture focused completely on the thyroid gland, and it should be clear, a lot of this entire lecture series is knowing and describing these organs and the various components to them, and that's kind of it. So you gotta really focus and get through all of these components. And so now we move on to the adrenal gland, and it's kind of the same setup. Fortunately, we don't have the breakdown of how any of those hormones are formed. All right, and if it wasn't clear, the point of describing how thyroid hormone forms is, again, teaching you another process on how various organs or how various hormones are formed. It's a fairly similar, it's going to be a fairly similar process where atoms are getting rearranged, because that's, of course, metabolism. But anyway, back to the adrenal glands here. So, of course, you should note the adrenal glands sit on top of the kidneys, and we broke it down into two regions, and at this point, you should know the adrenal medulla is where the catecholamines, or those tyrosine derivatives, epinephrine, norepinephrine, again, water-soluble hormones released during sympathetic stimulation, that's located in the adrenal medulla. And so if I do have a model in the classroom that looks like this, if I point specifically to this region, I'm looking for adrenal medulla, and if I ask what hormones are released from there, it's epinephrine, norepinephrine, and there's really no debating that. And then again, epinephrine, norepinephrine functionality is the emergency metabolic activity, metabolic activity under sympathetic control. So however you want to view that, that's what you should be looking for. But really, our big focus when we talk about the adrenal gland is focusing on the adrenal cortex. Again, this is where lipids are stored. So they're stored kind of in this region here. And we're going to talk about three main types, but really I would argue that there's two major types that we're going to break down. And so I've stressed over and over, this is the histology that you need to know. If you can break down the adrenal gland to these three layers here, and I guess four if we want to include the adrenal medulla. And again, if you know, aldosterone is secreted from zona glomerulosa. It's secreted when we have high potassium levels and low sodium levels, or when blood pressure is low. And what aldosterone does is it causes the kidneys to reabsorb sodium and push out potassium, and by doing that, water follows wherever sodium goes. So water follows sodium, and that helps reabsorb water. And so notice it's stimulated by angiotensin 2, which you know is part of the renin pathway. It's stimulated by elevated levels of potassium, stimulated by low levels of sodium, and then notice AMP and BNP inhibit this. And again, AMP, BNP are the only hormones that reduce blood pressure. So overall, can you note that the superficial high-density nuclei region is going to be called the zona glomerulosa? That's what you need to know. You need to know aldosterone is secreted from there, and you need to know stimulation of the uptake of sodium and release of potassium, which helps increase blood pressure. It's a hormone that is in response to low blood pressure or low sodium levels, which results in an increase in blood pressure. The low density of nuclei, middle region, you need to know is the zona fasciculata. And that's where we have our glucocorticoids, which I rarely refer to them as that. Again, it's cortisol or cortisone is the term I tend to use for the glucocorticoids. And note, this is targeted by most cells. Now, when are the glucocorticoids secreted? Well, they are secreted when ACTH from the anterior lobe is being released, and that ends up targeting, of course, this zone to cause the glucocorticoids. Really, it's going to be secreted under long-term stress, and certainly some short-term stress, but overall long-term stress is when the glucocorticoids or cortisol and cortisone are released. And what you need to understand is this is the when I call this is the stress metabolic activity. And what that means is our body wants to produce glucose because that's our main energy molecule, and it does so by breaking down skeletal muscle. Okay, and so releasing amino acids from skeletal muscle so it can form glucose, and then breaking down lipids as well to form glucose. Now, if you don't fully understand metabolic activity, what that means is our body is capable of producing glucose. So even if you avoid sugars, our body is able to produce it. And so this is saying that, hey, we're going to break down skeletal muscle, which obviously doesn't sound good. And even breaking down lipids, which is long-term energy storage, we're going to break those down because of stress response. And then there's usually an issue with the immune system, whether we have too high of glucocorticoids or even if we have low levels of glucocorticoids. So we do get this in a regular amount, but in most cases, it's kind of it's more on the negative side because it literally is forming glucose at the cost of muscle. All right, and then you need to know the medium density is zona reticularis. It's on the deeper side of the histology. And that you just need to know is androgens. Now, androgens, it's not something that I test too heavily on. You can see ACTH will also stimulate this release. But basically, this is going to stimulate some of the pubic hair development, i.e. just starting to develop some of the sex characteristics. It is a sex-based hormone. It's just not one that I stress too much. We focus on testosterone because it has more specifics. This tends to be a precursor or tends to be more geared towards being similar to like a testosterone precursor. All right, and then again, this layer that kind of changes in color and gets really, really dark and dense, that's going to be the adrenal medulla. All right, and so you need to be able to identify the adrenal gland, each specific region, a hormone from the region, and then the functions of these hormones. And if you can do that, you really need know this organ, and you're good to go. That's the main point, the main extent of what you need to be able to break down here. All right, and so again, it's kind of not the greatest example, but in this region up here where we have heavy density of nuclei, this kind of in these regions coming across here, it gets a little bit heavy. That's going to be, of course, the zona glomerulosa. Now, I don't know if I would necessarily use this. I definitely wouldn't use this for zona glomerulosa, but you can see zona fasciculata is pretty good, and it has a really dense zona reticularis down here, and then a pretty light sort of adrenal medulla. Very, very minor zona glomerulosa. This is an example where you can kind of see the layers, but really over here, this is a great example. Again, another great example here. We got high density, light density of nuclei back to medium density and a complete color change that gives you the medulla. Same thing here. These are all pretty good images. Actually, scratch that. This one I would not use. I just wanted to show you that some of the prep, you actually have low density, heavy density, and then medium density in some of the different preps. The preps that I test on are always going to be heavy density, light density to medium density. So expect this sort of setup or this sort of setup if you're studying. Look for them. That doesn't mean these are bad preps. They're just kind of different. They're not either there's something different with the organ. There wasn't a big zona glomerulosa, or it's a completely different prep style. Okay. Again, the main ones I focus on are those kind of kitty corner ones to each other. Now, you should note the adrenal cortex can experience some imbalances, and so we can have hyposecretions, which again are going to be low levels of secretions. Now, when we talk about hyposecretions of the mineralocorticoids, you should note that the mineralocorticoid, or again, aldosterone, and so having low levels of aldosterone, is going to lead to low levels of sodium, which is called hyponatremia, and high levels of potassium, because again, its goal, when we think of what does aldosterone do? It uptakes sodium. So we're no longer uptaking sodium, which means there's a reduction in the body. And since it's a bringing of sodium and then a release of potassium, that means potassium is going to be high. Now, there's acidosis associated with this. I don't really get into this too much right now about pH balance. We're going to focus that. Certainly have a lot of time to focus on those things. All right, but notice it lowers blood pressure, which should make sense. I told you aldosterone is often secreted when blood pressure is low. And then because now you have a sodium and potassium imbalance, notice tremors of muscles. But your main points that you would be focusing on if you're studying this is hyponatremia is going to need to be mentioned, or hyperkalemia. One of those two minimally. And then I would probably at least mention low blood pressure, because those are the major points there. Now, we can also get hyposecretions of cortisol. And that would occur due to the removal of a gland or potentially loss of a function of the gland. All right, or there can actually be an ACTH imbalance as well. So having like the pituitary gland have some issue as well. So in either case, a hyposecretion of cortisol is going to result. And what that, of course, again, cortisol's goal is to form glucose. So in this case, if we're no longer having the hormone that's going to produce glucose, we're going to end up resulting in hypoglycemia. It could be a problem. Now notice there's a depressed immune system. That's going to be a result of whether having too much cortisol or too low. Again, to be clear, we do use cortisol and we use it at an appropriate level, but we don't want to increase it. Okay, so throughout the day, we have cortisol levels, but it's not the main hormone that's regulating blood glucose. So here, when cortisol levels are in an imbalance, that can cause hypoglycemia. Pretty rare, but in this case, if it's an adrenal cortex balance, it certainly can do that. Now notice loss of appetite, nausea, vomiting, weight loss can also be associated, but the big one that I have to list is definitely going to be hypoglycemia here, okay? Now, Addison's disease, you should understand, is when there's a secretion in both. It's a low levels of secretion, i.e. the adrenal cortex has some severe issues, so none of the hormones are actually being secreted appropriately, or are being secreted at too low of levels, I should say. And of course, if we just combine kind of the effects here, we're going to have low blood pressure, we're going to have muscle weakness, that's all the mineralocorticoids. We're going to have we're going to have weight loss, so that's going to be low levels of cortisol. And then we're going to have constant fatigue because we're not getting the right energy, again, from the cortisol. So no Addison's disease, know that it's low secretions from the adrenal gland. So Addison beginning with an A, adrenal gland, you just need to know it's on the low side of both secretions. Now notice the androgens weren't mentioned because they're not one of the major hormones that I'm going to be focused on, because again, those tend to be more similarly to like testosterone and tend to be precursor that leads to sex hormones in general. So it's not one of the main ones that has significant functions in comparison to things like testosterone, where we have very definitive functionality there. All right, so focus on the hyposecretions of this. Know it's Addison disease when we have both, and then be able to pinpoint if I say hyponatrium and low blood pressure would be signs of which of the following. Well, it would be signs of hyposecretions of aldosterone, okay, or hyposecretions of mineralocorticoids. Now note, aldosterone can go here and be a correct statement. Similarly, now if we have hyposecretions, we can have hyper, and as we know, hyper is on the high side of things. And so we can have mineralocorticoids. Again, the other term could be aldosteronism. And notice this is typically going to be, again, caused by a tumor. So in this case, this is a tumor that causes an increase in function. And so, or we can say the disease aldosteronism. And so in that case, you're secreting too much aldosterone, which means you're going to have hypernatremia and hypokalemia. So too high of sodium, and then really low levels of potassium. Now notice when we use aldosterone, so notice aldosterone does have something to deal with losing hydrogen. And so in that case, we get blood alkalosis. In this case, where when we had low levels of aldosteronism, we weren't exchanging and getting rid of the acidity, which is the hydrogens. And so in that case, we were becoming more acidic. In this case, we're becoming basic. Notice high blood pressure in this case because we're continuing to bring in too much sodium. Notice weakness in muscles still. That's because we have an imbalance of sodium and potassium. So think sodium-potassium pump. And then notice acidic urine. So with aldosterone, if you want to add to your notes, which wasn't something that was stressed, but aldosterone does cause the release of hydrogen ions, or causes a decrease in your, or sorry, causes an increase in your pH. It gets rid of the acid, where notice here, we're having too high of levels because we're getting rid of too much acidity. Our blood is becoming basic. All right, cortisol, so having too high of cortisol secretion, notice this is often a tumor in the gland, and this can be called Cushing's syndrome. So no Cushing's syndrome, and this can be hypersecretions with androgens for the Cushing's syndrome, but we're going to focus on the cortisol. So notice, again, cortisol's goal is to create glucose. So notice hyperglycemia can lead to diabetes, but the main point that I would stress is tissue protein damage. All right, so know that. And then the other things that I like is distribution of adipose. Cushing's syndrome is really, really unique. It's pretty obvious people that have them. There's actually the bulging eye syndrome as well, those sort of things. But with this, because again, it's often associated with stress. So when your cortisol levels start to really rise, notice you're not only going to have euphoric feelings, but you can also have swings of depression as well. So very, very unique imbalance. So the adrenal gland obviously has some importance to it. Pineal gland really is knowing that melatonin is being secreted. At this point, you should know it's tryptophan derivative. I think this is one of the more direct and kind of simpler sort of topics, so I'm just going to move right along here. So notice the first lecture number 3 was really focused on your thyroid and your adrenal gland. So if you really break this chapter down, it's just breaking down each organ, starting with the hypothalamus to the pituitary gland and dividing the pituitary gland into an anterior, posterior. Then we go to the thyroid gland, pancreas, the adrenal gland. Can you break these organs down? And if you can, you're in great shape for the upcoming exam. All right, but let's keep moving along here. We, of course, are moving on to the levels of hormone interactions. These are really basic. These are definitions. What you see on here is what you get. You need to know at this point antagonistic is opposite effects, and then you need to know some of the examples. And at this point, PTH and calcitonin is a good one. Insulin, glucagon. For synergistic, these are additive effects. PTH, calcitriol. ADH, aldosterone to increase blood pressure. Those would be kind of the same things, so no synergistic. And then permissive, the only example of permissive I know is epinephrine. Norepinephrine requires the presence of thyroid to be effective. All right, permissive means another hormone needs to be present for the other hormone to function. These are all definitions. If you know the definition and then you know the combination, which, this was very similar to antagonistic muscles, synergistic muscles, those sort of things working together, working in opposition. Obviously, we're just applying it to hormones. So that should have been pretty basic. Similarly, these are some of the last major hormones you needed to know. So don't underestimate the importance of the kidney. I do test heavily on the kidney. And so, again, calcitriol, main point was similar effects to PTH by stimulating the osteoclast to break down bone and therefore increase blood calcium levels, as well as help with absorption at the intestines. That's the main point of calcitriol. All right. And don't forget, this calcitriol is one of the lipid-soluble hormones. We have erythropoietin. You should know that stimulates red blood cell production. We'll talk about this really when we get to chapter 19. That's where we talk about whole blood. But for now, EPO, you need to know, stimulates red blood cell production, and that's sensed by the kidney. So as blood flows through the kidneys, there's about a 20 to 25% output of blood flow through the kidneys, and so that your kidneys are regulating and making sure you have the right amount and right concentrations of red blood cells, especially if it's sensing low oxygen levels or hypoxic conditions or hypoxia. All right, and then renin, like I've said, you should know ultimately increases blood pressure, and it actually its release is stimulated by sympathetic stimulation. So if you want to put that, sympathetic stimulation causes its release. Also, low blood pressure causes its release. So notice it's all sensory related on what stimulates its release. But if you remember, it eventually increases blood pressure is the big picture function of renin. So in that case, if I ask big picture function of renin, it's literally just going to say increased blood pressure. It won't mention any of the kind of steps involved that are over here, which I'm about to get to. All right, intestines, just know they secrete hormones that regulate digestion. It's the only organ so far that we're really talking about. Now, again, pancreas helps with the absorption of the nutrients, but that's not regulating digestion. That's after the fact. Digestion means is the breakdown of molecules through the point of absorption. So don't think of the pancreas as regulating digestion. It does not. It helps with the absorption. Intestines regulates the actual digestion. Now again, renin's direct effect, and you can likely see this on a quiz or an exam, renin's specific effect is it converts angiotensinogen. This inogen ending, if you remember, means it's non-functional. It converts it to its functional form, angiotensin. So notice the inogen ending is gone, angiotensin 1. Now, you might have heard of ACE inhibitors that help reduce blood pressure. Well, renin is the first step in a chemical pathway, and it's one chemical being converted, so angiotensinogen being converted to angiotensin 1. These multiple steps allow our body to really regulate itself. So ACE is produced by, we're going to find out, the lungs, and ACE converts, it literally stands for angiotensin converting enzyme. It converts angiotensin 1 to angiotensin 2, and then angiotensin 2 in the blood causes aldosterone release, ADH secretion, and stimulates the thirst when it hits the hypothalamus, which ultimately leads to an increased fluid take in some way, right? Aldosterone is going to drive sodium back in, water is going to follow, ADH is going to stimulate more reabsorption of water, and then drinking something, of course, helps bring blood volume up. Definitely need to know this one. Renin is a big one, so don't skip the kidney. It's kind of an important one to really know. The heart obviously monitors blood pressure. You do need to know natriuretic peptides. It's the only hormone that decreases blood pressure. You're going to see this on many quizzes. So get familiar with natriuretic peptides. It's the only hormone that decreases blood pressure, where when we talk about the hormones that increase blood pressure, you have renin, you have aldosterone, you have ADH. Those are the major ones that are going to increase blood pressure. And you can even argue erythropoietin, even though that causes more red blood cell production. But if you add ingredients to the blood, it increases blood pressure as well. Okay, so definitely note this. We're going to see natriuretic peptides quite a few times. Okay, thymus at this point, you should know, is the site where T cells mature under the influence of thymosins, right? Thymosins was the hormone that stimulates it, and it is specifically the thymus that produces it. So T cells mature at the thymus. That's why they're called T cells, thymus derivative, and it's specifically the hormone thymosin that converts them to the actual T cells. Again, that's a connection from the previous some previous lectures, so you definitely need to note that. All right, testes, estrogen, you pretty much need to go through this. Testes obviously are going to lead to the production of sperm. That's what spermatogenesis is, and the development of secondary sex characteristics. Estrogen, again, secondary sex characteristics, but really follicle and egg development is what I stress for now. Progesterone, you should know, is what maintains the uterus. And then again, it does help with mammary gland development, but mostly what I focus on with progesterone is maintenance of the uterus. All right, just know those. Those are pretty much facts and things that you need to have memorized. All right, and so the final concept was general adaptive syndrome. This was our stress response. As I said, cortisol levels are maintained at a pretty low level in the body. Like I said, our body does go through some stressful parts throughout the day, and that cortisol is important for adjusting to those. But really, when we talk about our stress phase, usually the minor everyday stress is actually epinephrine, norepinephrine. And so you should know our alarm phase to deal with everyday stress is really that shot of epinephrine, norepinephrine, which only is a short-lived response, right? Lasting a couple hours, something along those lines. All right, so no, alarm phase, epinephrine, norepinephrine, sympathetic stimulation, that's our everyday stress. Associate those terms and you're good there. Then you should know the resistance phase is the second phase. That's when stress is longer than a few hours, lasting probably days to weeks. And basically know that this is where we start using the steroid hormones from the adrenal gland, i.e. your cortisol. If cortisol is not clear from this, write on your notes, cortisol secretion. So in this case, we're maintaining glucose, and hopefully this is a short-lived sort of response, but we're ending up depleting some of our amino acids and muscles and some of the tissues. Now, if this maintains for long term and we're constantly breaking down muscle tissue, it leads to the exhaustion phase, which means there's actually damage to the organs. Okay, so I would have to mention something about loss of all reserves and damage to organs here. In this case, I would be mentioning the use of cortisol and the loss of a lot of your energy reserves, all right, such as your long-term energy reserves and fats. All right, and so this is really definition, so don't overthink the general adaptive syndrome. Now, the final part to kind of wrap this up, we have the hard concept here, but we've been stressing these, so I'm hoping you've realized, and hopefully going through the review at this pace, you sort of realized how many things did actually overlap and how many concepts sort of lined up. So I'm hoping you see that. It is very hard to see in a class that moves, especially this pace, but even at the 16-week mark, it's a tough concept. So we're trying to get you to grasp the big picture concepts here. All right, so we talked free versus bound hormones, and again, when you talk free hormones, you needed to know, hey, these didn't need a protein. These are going to be the water-soluble hormones because they're unattached, and so we, of course, are talking about peptide derivatives, the catecholamines, and the tryptophan derivatives. All right, these tend to remain functional for less than 1 hour. And that's because enzymes can break them down. Bound hormones, these were those that have to attach to a transport protein. We really talked our lipid-soluble hormones. Again, your reproductive hormones, the hormones from the adrenal cortex, and your thyroid hormones tend to be the main ones, along with calcitriol. These are going to be remaining functional for longer periods because they're bound to something else, which makes an enzyme harder to break them down. But regardless of how long they're lived, really two processes occur. They either bind to their target cells and cause the effector to actually perform its function, or they get broken down. And that's the main point, is you're going to start seeing the liver kind of breaks molecules down, rearranges the atoms, and then the kidneys get rid of that through your urine usually. All right, or they can be destroyed by enzymes in the blood plasma or within interstitial fluid. So, and I remind you to think acetylcholine esterase. All right, but what happens, or which hormones bind intracellular receptors? Well, again, those are the steroid and thyroid hormones. And really all you need to get from this, and I know it's really complex, I say all you need to get from this, but the main point is the way hormones function is by activating genes. The difference is steroid hormones and thyroid hormones, they function when they bind to intracellular receptors, and then they move and actually form a hormone receptor complex that starts or activates gene synthesis. That's all there is to this. Literally, hormones stimulate their target cells to start the gene activation in some way. All right. For steroid hormones, that's as simple as we leave it. Binds intracellular receptors and activates genes. For thyroid hormone, it does this exact thing, but the other thing is, notice it has a receptor on the mitochondria, and if you remember, the function of thyroid hormone is to regulate metabolism, so ATP production, so that's one of its function as well. All right. Main points, lipid-soluble hormones function by binding intracellular receptors, which often results in gene activation. The exception is thyroid hormone can also activate the mitochondria to produce ATP. That's the big difference, but if you can walk away with kind of that big picture, excellent job there. All right. Now, the more complex one was, of course, the water-soluble hormones. All right, and that's because they're not lipid-soluble, and since that means they don't bind intracellular receptors, that means there needs to be an internal chemical cascade that occurs. And so the first messenger is always going to be your peptide-based hormone or your water-soluble hormone. Your second messenger tends to be the one that actually carries out the response. Regardless, the major point here is peptide hormones work by actually activating a G protein. 100% guarantee I ask about G proteins. You need to know these are not second messengers. These are chemical intermediates that activate second messengers or activate the chemical cascade that leads to the production of second messengers. This is an intermediate. It is not a second messenger. Make sure you know that. 100% guaranteed on the exam that you need to know what a G protein is. All right, second messengers, these are going to be the molecules inside the actual cell that trigger a response. But notice most of the response is either going to activate genes or increase or decrease metabolism like we've already seen. All right. When we talk about this is how the cells can respond. And so the way the cells can kind of limit a response to hormones is by adjusting how many receptors the cells are presenting on their surface. And so downregulation is a decrease in the number of receptors, and therefore it's a decrease in the response. And you can think diabetes, specifically type 2 diabetes, which is insulin resistant. It's insulin resistant because the body got so familiar with high levels of insulin that the cells said, hey, we got to stop this high response. And so they decrease the number of receptors, and therefore you are now considered insulin resistant. Upregulation, of course, is the opposite effect. That's going to be increasing the number of receptors to generate as a response. And this happens, of course, during a woman's cycle. Luteinizing hormone receptors increase right around day 14, which stimulates ovulation. So that's an example of upregulation. Now, these are just kind of background information to tell you how the cell itself can respond to things. All right, but we focused on really calcium as a second messenger. That was one of them. And really what you needed to know is that it eventually led to the production of calmodulin. And what calmodulin did was activate enzymes. And those enzymes eventually are usually going to lead to the final production of a protein or are going to activate gene synthesis. It really doesn't matter. This activation of enzymes is activating a enzymatic cascade that leads to the final product. So it could be something like insulin release, like there's insulin exists as proinsulin, all right, and so maybe when there's a signal, it causes the release of insulin and all those chemical changes. All right, so all you need to know is when calcium enters, it stimulates calmodulin, which activates enzymes. When enzymes are activated, that's generating a response. It's one of the things that our body does to, again, regulate internal conditions, is it changes the enzyme concentration. So if we need a great response, have tons of these enzymes available, and that's going to generate a large response. Or if we need a short response or not such a dramatic response, there won't be as many enzymes. Even though there's a signal being sent, there won't be as great of a response. So notice there's a lot more going on than just what we've covered. It seems like we're covering a lot, but we're really only scratching the surface of the endocrine system because it's quite complex. But again, main function of calcium is it activates calmodulin, which activates enzymes. All right, and then you needed to know that if if our G protein activated the enzyme adenylate cyclase, you needed to know that that led to an increase in cAMP. And if we have an increase in cAMP, we basically activate kinases, which, again, it's an enzyme that activates other molecules to basically activate them. That's it. So cAMP leads to activation of kinases, which activate other molecules. Notice, activating enzymes is key. And when we activate enzymes, as I continue to stress, it's just part of the chemical cascade to lead to a final product. So if you see adenylate cyclase, you should just think cAMP is going to increase. What's the point of cAMP? It activates enzymes by activating kinases. What's a kinase? It's an enzyme that phosphorylates another molecule. Okay, I literally studied how phosphorylated a specific molecule was to determine how active it was. All right, that was some of my research. All right, but the main point, adenylate cyclase, you think cAMP goes up. Otherwise, if we have a decrease in cAMP, the way that happens is the activation of phosphodiesterase. So notice, depending on the enzyme, this was what I was saying earlier, the internal or intracellular enzymes or cytoplasmic enzymes, those matter. In this case, if this was the same, notice epinephrine, norepinephrine are examples for both. When adenylate cyclase is available, cAMP goes up. When phosphodiesterase is available, it's to lead to a slower response. Because remember, epinephrine, norepinephrine are the sympathetic hormones. So under parasympathetic stimuli, so things like organs that are under parasympathetic stimuli, epinephrine would be causing inhibition to those. All right, so phosphodiesterase, the main point here is phosphodiesterase is going to reduce cAMP, and by reducing cAMP, we have less enzymatic activity, which means we have less going on in the cell. All right, this material is, again, one of those concepts that really separates the A to B student there. It's certainly the more complex things. If you had a decent understanding of it, great. It's hard to break it down as simplistic as I did because it kind of seems like it's too short of material, but that really is the main point. Calcium stimulates calmodulin. Calmodulin is going to activate enzymes. That's calcium as a second messenger. If our G proteins activate adenylate cyclase leads to cAMP. cAMP as a second messenger is going to, again, activate kinases, which phosphorylate or activate other molecules. On the flip side of things, when G proteins activate phosphodiesterase, that is actually an inhibitory response, breaking down cAMP, showing you that cAMP is an important second messenger that drives responses, where adenylate cyclase increases cAMP, phosphodiesterase would break it down or slow it down. All right, but that is the full review. It took me almost an hour and about an hour and 20 minutes. So quite a chapter to get through. This definitely was the harder side, but if you really think about this and if you wanted to know your main focus hands down, it's really breaking down each organ. And I would just focus on breaking down an organ at a time. Start with the hypothalamus. If that's too complex, then maybe go to the thyroid and kind of work on the stuff that has really in-depth lab stuff, because of course the lab exam is going to happen first, the lab content. So get familiar with lab concepts first, and then the lecture stuff should fall a little bit easier. As usual, if you have questions, please do not hesitate to bring those to class. I look forward to seeing you in person, and I hope this worked out well for you. I hope this gave you a nice overview, and I hope most of the lecture you were kind of nodding your head, yeah, this is familiar, yeah, this is familiar. And I hope that I pointed out some of the areas where you might have struggled. Definitely the final content was quite challenging. All right, look out for Chapter 22's version of this, and that'll close out Unit 1. Take care, everyone.